do our fourth and final talk before we do our panel session. Our last speaker is Dr. Ed Kelly. He's a professor in the Department of Pharmaceutics at the University of Washington and part of the Transporter Elucidation Center at their university. His lab utilizes complex in vitro models, also known as New Approach Methodologies, or NAMs, to study drug and xenobiotic disposition and toxicities. And some of his notable current activities include working with the FDA's iSTAND program to qualify a new kidney NAM as a drug development tool. Welcome, Ed.
Well, thanks for the introduction, Lauren, and thanks for the organizers for the invitation. So I'm going to wrap this session up talking about the intestine. So first off, I want to talk about the UW Tech Transportation Center. So the co-PIs, Dr. Jashu Nadkat and Dr. Joanne Wong, and I'm a co-investigator on AIM-3, looking at the developing intestine, and Dr. Sam Arnold is the co-investigator on the proteomics. So we have Dr. Nick I, who is the guy who gets us placentas from Madigan Army Hospital, and also all the folks down below which do all of the work. So, human enteroid monolayers, I would say, are a promising complex in vitro system. The industry standard, let's see if I can get this, like you got it. Industry standard for looking at drug transport are the Keiko-2 cells. Okay, so Keiko-2 cells, when you put them in what's called a Transwell system, so it's a simple in vitro model, they will form tight junctions, and so then you can look at from a basolateral to apical or apical to basolateral vectorial transport, and you can do that very well. Unfortunately, Keiko-2 cells, when it comes to the intestine, they're kind of like the Hep G2s of the intestine. For those of you that are liver people, Hep G2 cells are a liver cell which don't really express many P450s. Keiko-2 cells are the same sort of type of cell, not making a lot of P450s. My colleague, Dr. Ken Thummel, retired last year. He's really interested in drug metabolism, P450s in the intestine, so he uses LS180 cells. Okay, LS180 cells have good CYP3A expression. Okay. But unfortunately, they don't form tight junctions. So you put them in a Transwell system, and you can't look at vectorial transport. Okay, and in both cases, Keiko-2 and LS180 cells are cancer cell-derived cell lines, and so they of course have their inherent issues there as well. Okay, so then there's things like primary enterocytes in suspension. Those are things which actually they're commercially available, but they have a very short half-life in cell culture because they are the terminally differentiated cells. So then there's iPSC-derived organoids, which iPSC-derived organoids have the potential lasting forever because they are pluripotent stem cells. However, as we just learned from Dr. Lamp, iPSC-derived cells really are only going to get to be fetal in nature. So it's like, as I said at lunch today, iPSC-derived cells, they look like a dog, they bark like a dog, but they're actually more like a fetal puppy because they don't have that full phenotype. So what we have been doing for the past few years is we have been working with human enteroids. Okay, so human enteroids are cells which derive from the crypt cells that exist in all of our intestines, and they're self-propagating, so we can work with those, and they have all the properties that we're interested in. In Transwell systems, they will form tight junctions, they will have transport function, they will also have things like the P450s, and also you can derive them from different sections of the intestine, so you can recapitulate the zonal expression that you see in the small intestine. Okay, I have to show this slide because I love this slide. This is one of these slides, one of these experiments we could probably never do in modern day IRBs. But this is some work that was done by Dr. Ken Thummel with one of his former graduate students, Mary Payne, who's now professor at Washington State University School of Pharmacy. But they were looking at the intestinal extraction ratio of midazolam, which we just heard about from Jed again. And so how did they get the intestinal extraction ratio of midazolam? So what they did is they had folks who were undergoing liver transplants, and while they were anhepatic, they then administered the intestinal arterial vein midazolam, so that from that they could actually look at what was the extraction ratio only from the intestine. Okay, so very interesting, very amazing experiment. When we take our same intestinal enteroids, put them into a Transwell system, and then say what is the extraction ratio, and I'm from the School of Pharmacy, so I have to show at least one equation here. But what we see here is you get the in vitro midazolam extraction ratio from these Transwells is almost right on spot with what you see in vivo. So it's a very good model system for recapitulating something which is a very complex function. Okay, so the anatomy of small intestine, everyone knows we get three sections, duodenum, jejunum, ileum, and there's the villus here. So the villus is what's important because it contains in the basal layer, the crypt, is the crypt cells. So the crypt cells use LGR5 positive multipotent stem cells. So how do we make intestinal enteroids? So what we do is we'll either do biopsies or whole human tissues and get out those crypt cells and propagate those in media that has high levels of growth factors, okay? And we take those and populate them with high levels of growth factors. What you'll get is these, what we call these spheroids. And so these use LGR5 positive, undifferentiated crypt cells. You then take those and you withdraw growth factors, in particular Wnt3a. When you draw that Wnt3a, what happens is they will differentiate and they'll form these involuted structures that have not just the enterocytes that are at the tips, but also the other cells like goblet cells and Paneth cells. And there's very beautiful structures here like this, but we're really interested in looking at transport and uptake and things like that. And these enteroids like that, after you differentiate them, the inside of that enteroid is actually the apical surface. So unless you have some way of getting drugs inside of these structures, which are like 50 to 100 microns apart, it's not really feasible. So what do we do instead? So what we do, and actually this is some background for our UW Tech I'll tell you about, but what the workflow is we're looking at tissue profiling of transporters, in particular SLC and ABC transporters, and to make this in vitro model, what we're working with here is we're looking at the ontogeny. If you want to look at first or third trimester, you can take iPSCs and differentiate them first for like a month in vitro, and they look like a T1 intestine based on single cell RNA seq. And this is archival data. If you take those same cells, differentiate them further by in vivo propagation, and then look at them again in vitro, they look like a third trimester. At the UW Tech, what we're really interested in is looking at neonate and pediatric donors. So what we're doing is we're getting from organ procurement organizations whole humans' intestines, and then generating these spheroids that we can make cell lines from, and then taking those. And what we do is we take those intact enteroids that are differentiating, and we essentially break them apart and put them into these transwells where they form a 2D monolayer, which allows us then to look at that vectorial transport. And guess as this is not the right audience for this, because everyone knows this, but development is dynamic. So if we look at, say, for example, an induced pluripotent stem cell versus a T1 human intestinal organoid, looking at SLC transporters, which are important for drug, nutrient, and xenobiotic transporters, we see, of course, dramatic changes. If we look at first versus third trimester, over 234 differentially expressed genes just for the SLC transporters. And if we look at, for example, a human intestinal organoid that's undifferentiated and then differentiated in vitro and put into a NAM, we then see about 60 differentially expressed SLC transporters. Okay, so what we've been doing as part of the UW Tech is building this library of donor cells. And so what we do is we'll get in whole human intestines, and so we'll get, ideally we'll be getting duodenum, jejunum, and ileum, and then we'll take those and have the intact tissue to then look at transporter profiling by proteomics. And in addition, the goal is from each one of these sections to make cell lines as well, okay? And so I want to focus here on, what I want to show here is like we have a diversity of donors we collected for the past 2 years, ranging in age from 8 years old to 5 days old, excuse me, and also from both sexes as well as a variety of races. So a diverse panel that we're generating here. Okay, so why is measuring protein abundance so important, okay? Well, we all know that, but I want to show you here is that this is looking at 3 different probe substrates for CYP3A4 across a variety of different donors. And what you see here, it looks like a very good correlation in the top panels between mRNA in the open squares versus protein levels in the closed circles. But what you see is when you look at the low levels of expression in certain donors, that correlation between mRNA and protein falls off dramatically. And this is not surprising. This has been seen in many different studies for many different tissues. So it's not a surprising thing to see. So that's why we're focusing in on looking at the proteome. And so how are we doing this? So we're looking at data-independent acquisition, so-called shotgun proteomics, to look at the entire proteome, okay? And this is the workflow. This is from Dr. Arnold's lab, because he's doing both DA proteomics for both the placenta and the intestine for the UW Tech. And I'm not a proteomics person, but basically the flow is the same in that you get your tissue, you process it, make membrane preps or whole cell lysates, and you run it on the mass spec, and you're generating an in silico library to actually look at what are the different peptides that correspond to the different proteins. And this is in contrast to targeted proteomics, which is much more quantitative. So why are we doing DIA proteomics versus targeted proteomics? With targeted proteomics, you'd have a peptide standard for each and every protein that you want to quantify. So in the case of the SLC transporters, there are literally hundreds of SLC transporters, and peptides are not cheap. So we're doing the DI proteomics and looking kind of like at relative expression levels. And so this is some data looking at across mucosal regions. So this, when we get the whole intestines in, we basically scrape out the mucosa, and we use that to isolate protein. And this is looking at the mucosal regions, across age groups as well as regions. And long story short, you don't see a lot of differences in terms of the age ranges, but you do see, certainly in the adult, you see differences between the duodenum, ileum, and jejunum. And this is not surprising because this is known, and this is the entire proteome. So it's not just the transporters. And then we looked at transporter expression of mucosal regions of age groups. And this is the kind of slide I always tell my students, don't put up a slide with so much stuff on here to see because it's so difficult to read. But bottom line is we didn't see a lot of differences, okay, in mucosal regions, okay? So, but we're really interested in not only the mucosal regions, but looking at this from a NAM perspective, okay? Why are we interested in looking from a NAM perspective? Well, a lot of different reasons. And one is that, with the cell lines, we can actually look, those are going to be archived, we can look at them in many different times. The other problem is when you're looking at the mucosal regions, we're not down in the OR harvesting intestinal tissue. These are coming from organ procurement organizations. And so we get a call and say, we've got this available, are you interested? We'll say yes, let's have the surgery scheduled, they'll put it in UW solution, courier will deliver it to us, and we're talking about something like, anywhere from 20 to 36 hours later. So there's always that caveat of what is happening to transporter protein and mRNA expression in that time frame after surgery. So the other things that we wanted to look at was, let's just focus in, since this is a NICHD funded UC2, let's look at the small intestine mucosa just for our neonate and pediatric population. So what we have here is we were looking at, in this case, we were looking at both mucosa from either enteroids in Transwell versus enteroids in the mucosa. And we looked at, again, neonate, so 0 to 1 month, versus pediatric 1 month to greater than 1 month to 8 years. And again, what we saw, we detected over almost 4,000 total proteins. So good sampling of the proteome. But we really didn't see, again, a lot of differences in this case in terms of unique expression of, in this case, SLC transporters. So for example, focusing here on the enteroids, we saw 2 unique SLC transporters in the pediatric enteroids in Transwell versus the neonate. And again, if we look at the whole mucosa, we see 3 unique SLC transporters in mucosa of pediatrics versus the neonate, okay? And then we started looking at categorization of transporters, SLC and the ABCs in terms of functionality. And so, for example, here we saw a total of 14 ABC transporters in these samples here. Whereas if we look here, this is looking to SLC transporters, and this is a very busy slide again. So let's just focus in on what about significantly higher expressed SLCs in Transwell monolayer versus mucosa. And this is actually the pooled neonate and pediatric samples. So here we saw, in this case, 30 higher expressed SLC transporters in the Transwell monolayers versus what we saw in mucosa samples. And so, for example, we see a large number of ones of intracellular organelle transport. We see ones involved in carbohydrate transport, several here involved in metal transport, which I know the folks at Intech are interested in, as well as other groups. And so definitely saw some interesting things there. Okay, but what about enteroids versus Transwells, okay? So a lot of people still use enteroids, and these are actually a really good system for doing things like disease modeling. So for example, you could make Transwells from cystic fibrosis patients and look at how they respond to potentiators and correctors. So it's a very good model system for disease modeling. But again, like I said, we're part of the transporter elucidation network. We want to look at transport, so we want to look at what is going on here in the monolayers of the Transwell versus what is going on here. And so what we did is we took, in this case, we took a single donor, 5-day-old donor, and we looked at undifferentiated spheroids, so those LGR5 positive crypt cells. We looked at differentiated enteroids, so those cells that we differentiated kept those intact 3D structures. And we compared that to undifferentiated enteroids that are plated out in a monolayer versus differentiated enteroids plated out in a monolayer Transwell. And so this is just a workflow here. So basically we get in whole human tissue, we grow them in these matrigel domes here, and here you can see these undifferentiated, they look like these little balloons of cells, it's single monolayer cells. We withdraw the Wnt3a and we plate them out into a Transwell. We also do some stuff in 96-well plates. And what you see here is you see this nice swirling of the cells, and that's compared you just come down here, monolayer morphology. So here's undifferentiated cells and transwells, and there looks like a much more uniform surface, and there's also the occasional undifferentiated intact spheroid versus the differentiated transwells, where you see much more undulating type of morphology in these culture conditions. And again, we had 4 different variables: differentiated versus undifferentiated, transwell versus 3D. And this PCA plot, you get beautiful, 4 sections of these 4 different groups. And then if we just look down here in the lower right, just compare differentiated spheroids versus differentiated transwells. We saw almost 4,000 differentially expressed genes, so dramatic differences between culture in these intact 3D structures versus in the transwell monolayers. So in conclusion, one thing I want to let it be known that UW Tech is building a repository of neonate to pediatric intestinal donor tissues, as well as generating these cell lines. Again, these are cell lines that will be available as part of your sharing plans that you write into all our grants, right? So these are going to cell lines will be available. The majority of the proteome is similar between neonate and pediatric intestine. Comparing neonate, pediatric, and adult intestinal mucosa, we did see a few SLC transporters which were uniquely expressed. When we compared pooled pediatric and neonatal mucosa versus transwells, we identified differentially expressed both ABC and SLC transporters, suggesting that there is definitely a difference between that mucosa versus a transwell. And is that really due to degradation that occurs in the mucosa from that time from harvest to delivery versus when we recover it when we differentiate them in that in vitro model? And finally, the preliminary RNA-seq results suggest significant differences between intact 3D enteroids versus transwell monolayers. And from a transporter perspective, the differences we see are dramatic and suggest we should not be doing 3D enteroids. If you want to look at transporters, you definitely want to be doing transwells, not intact 3D cultures. And I think with that, I want to acknowledge all the folks here in UW Tech. So here's our group here. Joanne and Josh, the PIs, Sam Arnold, the co-I. Really want to put a shout out to Dr. Ching-Cheng Mao. He passed away a couple years ago, but he was the one who really, working with Dr. Nadcat, was the one who wrote the majority of this grant, and he passed away before it was funded. But that was a sad story there. Also want to acknowledge all the other folks that are involved in this. And with that, I guess we can open it up for questions.